Τι είναι το γονιδίωμα? Μπορείτε να το φανταστείτε ως ένα τεράστιο...
What is the genome? You can imagine it as a huge, infinite text of biological data written with 4 letters only, A, C, G, and T. This text for humans is about 6.4 billion letters, half of them coming from the father, the other half from the mother. All the script of our lives is included in there. Gender, development, intelligence, weight, height, hair. And, well, in this chapter, personally, well, I wouldn't mind changing a couple of pages. And also the predisposal for certain diseases. It is certain that knowing genomes better and technology evolution will change, has changed, and will change even more in the future medical practice because it offers more treatment opportunities, multiplies choices, but also increases dilemmas. How will we manage in the future this tremendous knowledge? Stylianos Antonarakis in Athens Festival this year is an Emeritus Professor of Genetic Medicine in the University of Geneva, ex-President of the International Organization for Human Genome. He will analyze challenges in the future and how the involvement of infrastructure of genomes will change medicine even more, starting and ending with the letter A. The conversation will be coordinated by Vasiliki Michopoulou, biologist and science journalist.
Καλή σας μέρα, καλώς ορίσατε.
Good morning, welcome to Athens Science Festival. I'm Vaso Michopoulou, biologist, science journalist, and today I will present to you a pioneer in the genetic medicine, Mr. Antonarakis. You're in Europe, though a little bit far from us. We're very happy to have you with us. Good morning, Ms. Michopoulou. Thank you for the invitation. Before we go ahead with your presentation, I'd like to say a few words about you, though I think that people know you. So, Professor has a big research field in genetics with lots of discoveries, with lots of enthusiastic discoveries. He started in the USA in the molecular basis of genetic diseases such as schizophrenia or Down disease, etc. He has studied a chromosome 21 in a human genome project that he started in 90s and completed in 2004. He has read, I think, his own genome. We know about 4,600 genomes connected with diseases from the total of 20,000. So there is a lot of unmapped DNA. He started monogenome diseases and unknown diseases investigation in Asia. He has been awarded on numerous occasions and has impacted the whole world with his articles. He's a member of the Academy of Sciences. He's the only Greek genetist with the biggest award of genetics in 19. He will talk to us about the genome, the ABC of life. Whatever we say about you, I don't think is much. There would be a lot to say about you and all the awards you have received. Thank you, thank you. Thank you, Ms. Michopoulou, for this great introduction of yours. I would also like to thank all the organizers for this fantastic event. I would also like to wish good morning to all the audience. I don't know if you can see my slides well. Well, again, acknowledgements for this great honor. And I'd like to thank the hundreds of people who have impacted and marked my scientific journey. I'll take you a few years back. Allow me, in 2007, it was a year of elections for Obama and McCain, the senators, and two colleagues, a biologist and a bioethic professors wrote that as of now, we should know, we should have information about parts of the candidates' genomes in order to vote knowingly the person who will have the biggest force in the world. Now, many years after that, maybe at the end of this small presentation of mine, you'll be able to understand whether we are ready and we have the knowledge to do that. So, as you said, and as the colleague that started this conversation said, I'm going to talk to you about the genome, this huge text of information that we have in those trillion cells of ours, with only 4 letters, written in 4 letters. As we said, when I finished medical school in 75 in Athens, genome was something unknown. So the era of the genome is of my generation. So the program of a human genome that started in the 90s was something huge, grandiose as a program. We had to read something unknown. We knew it was there, but we didn't know how it looked like. We wanted to map, to clone, to read the genome and see its variations. I'll come back to the variations. End of 2003, we read the biggest part of the genome, 95%. There was a big celebration in the White House with Clinton as a president at the time. We're again and now in 2021, and the difficult areas of our genome, this 5% is still not read. I'll give you a page, a picture of this book, the book of our genome, which is the most important book of life written so far. You see here about 10,000 letters of my genome, which is 320,000 bigger, 6 billion letters. And you can fit it in an iPhone, because an iPhone has about 16 million bytes. So your genome and your parents' genomes can be saved on your phone, just to understand the evolution of bioinformatics. As you said, Ms. Michopoulou, I have been a serious researcher of chromosome 21. You see it in the small circle here. It's one of the smallest chromosomes, so 1.6% of the genome, and it was read in 2000. I wanted to have a way to show the genome to people who come to our clinics with genetic diseases and to the students. I convinced the Swiss Organization of Bioinformation to print the book of chromosome 21. You see it here printed. It's about it weighs 4 kilos, 43 million letters, the size of font 8. We usually write at font 12. And this is 1.6 only of our genome, and the whole genome is 80 such volumes. So you can understand the extent of this genome and how easy or difficult it is to read the changes in the genome of each and every one of us. As we heard, it's infinite with a beginning and an end. So with the help of computers and the progress of bioinformatics, we can actually study it. So each and every one of us has a part of about 140 volumes of this encyclopedia from the mother, and the other half comes from the mother. So when we want to make a genetical experiment and to create a new life, we take a half genome from one parent, the other half from the other parent, and we make the new genome. Be careful, there is one very important element here. Each time that we copy the genome from one cell to another or from one generation to another, this copy, although it is very accurate, makes a few mistakes. So if we start with a genome that's in your left, my genome, that is when I was the zygotic first initial cell, every time it is divided and copied, there are some mistakes, such as this A that you see in the first division, and then some more in the second division, some more in the fourth, fifth, etc. So this percentage of mistakes is really statistically insignificant, one every 100 million letters, but since we have 6 billions each one of them, you have at least 60 different mutations from the father's and mother's genomes. This makes us polymorphic or diverse. And in this page again of my genome, I show you in flags here the positions of the letters that are different from the reference genome. One out of 1,000 is different between two random genomes. If you take the genomes from the father and mother who are not related in any way, you'll see that one every 1,000 letters is different. And what is the importance of that? It makes us different and unique. You can't have two people on this planet with the same, precisely the same genome, apart from monogenic twins, that when they start their lives as zygotes, they have the same genomes, but as they grow, they have new cells, body cells, and they decline one from another. So individual genomes, since we're all kinds of Homo sapiens, is 99.1 similar, but 0.9 different. So each one of us are a unique personality on this earth. So this differentiation of 0.9% makes us different one from another and have this admirable uniqueness that makes us different in our appearance, but also in all other characteristics. This diversity is a big gift of nature to us because it allows us to adapt and to evolve in the constantly mutating environment. If we're all the same clones, copies of one over another, every time that we would have an unfavorable change of the environment, we would all extinct. But since we're not all the same, every time that the environment changes, right, now with the appearance of a coronavirus, there are some people with genomes that are not affected by coronavirus. Some others are affected. So our genome is constantly in battle with the environment, but due to this diversity, we can evolve, adapt, and survive. However, in life, nothing is given for free. We pay for everything. And the secret, the obvious secret of medicine is that sometimes mutations, individual, particular mutations in each and every one of us, some of them are so rare that make us sick with genome diseases. This is the price that we pay for this possibility to adapt and evolve. Medicine, and we should understand that medicine is based precisely on that, on the fact of that we have a diverse genome, but sometimes this diversity makes us sick. And as Theodosius Dobzhansky said, the big biologist, nothing, we understand nothing in biology apart from the light of development. We do not understand medicine, that is, unless we see it under the light of evolution. Now, about our similarity with all other living beings, DNA makes RNA. We talk a lot about RNA because there are many coronavirus vaccines made with RNA. It codifies, it transcribes, if you want, and expressed in proteins that are structural units of cellular life. And proteins are built with 20 letters, DNA 4, proteins 20 letters. I have a picture here of a protein, which I love a lot, DIRC1A. And I love it because it's very important for the Down disease. Some letters you can see in the first line and underneath for the humans, and then you see the protein in various types of animals, the dolphin, the pig, the cow, the horse, till the fish. And you see that this part of protein is the same, is identical to all these species that I have here. And this is the biggest proof of the theory of Darwin, which is not really a theory, but it's the reality that all living beings on Earth have one same ancestor. This is the reason why we have a similar DNA and proteins. So medicine, after reading the genome, has become a genomatic medicine based on genome diversification. Some people call it individualized or precise medicine. Keep the term genomatic medicine or genome diversity because it helps us diagnose, prognose, prevent. And now genetists have a new tool, a new system that makes them specialized, the genomes. You know, cardiologists have the heart, neurologists, the nervous system, dermatologists, the skin, genetics, medicines, society have a very important new organ to study, which is the genome. If we consider that, well, medicine is something rational and not an historical evolving science. On this tree of medicine, there are two big trunks, the genome and its diversity, and the environment. These two different diversities. There are no diseases that are either one or the other, or an interaction between the two. All diseases start from there. Now, if Ms. Michopoulou, if one of you asks me to read your genome and give you my opinion, medical opinion on your genome, how from these 6 billion of genome I can give you one medical sentence. Only a small part that you see here, 0.35, is the one that I can explain. All the rest is unknown territory. We know how it is written, but we don't know its importance because, as Michopoulou said, about 667 to be accurate, is known to cause monogenomatic diseases. What are those? There are diseases that are there because there is only one mutation. One letter out of this 6.4 billion has changed. It can cause a vertigo. One letter out of 6 billion changes, and somebody cannot become tall. This is a characteristic picture of the skeleton of the spine. The genetic determination is absolute. Everybody with this change has this dysplasia. Same. We may have a mutation in these two nucleotides in a genome called the BRCA1. Then there is a possibility in the length of life, 70%, to have breast cancer. Without this mutation, otherwise the risk is 10%. So it's quite an important determination. If I can show you a video here to show you the importance of a genetic mutation and what happened because of it in the life of this footballer. Videos are not translated.
Εκεί που ήταν φυσιολογικός και έπαιζε.
He was normal, he was playing as a healthy person, but his heart stops. Sudden death coming from hypertrophic cardiomyopathy. It's the mutation of one letter in one genome called MYBPC3, but it's so serious that can cause a sudden death. And finally, about this small determinism, there is an APOE gene. It comes in 3 types: green, 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 red, and red, red. So if you are red, red, depending on the amino acids in the positions 112 and 158, and in the 2 genes from the mother and the father, has red and red, has a 12% more risk to develop Alzheimer's disease. This is a small determination, but it's very important to health. So in order to be able to help people with genetic diseases, we created in Geneva one clinic, the first called the Genome Clinic, with members from, well, it's a multidisciplinary medical clinic, bioethics, biologist, public health, etc. And after we read the gene of people with possible hereditary diseases, then we may provide a diagnosis and sometimes provide or suggest a treatment. So in our clinic, we have 6 big parts: molecular analysis, saving access in data, bioinformatics, a think tank, the clinical and consulting genetics. The think tank meets once a week in order to try and codify the knowledge. And there is the last part, the ethics, legal issues, and financial issues. Now, what is the unknown part? I told you that I can give you a medical opinion based on the 0.35%. The other part, which is bigger, out of the 20,000 genes that codify proteins. When I started in the 80s, we only knew one gene, the B hemoglobin that creates the thalassotherapy. Now, in March 2021, we have 4,660. We expect in the future for this red part, the unknown but predictable genes for genetic diseases with a dominant character. We need only one mutation to be sick. And the green part, which is about 7,000 to 8,000, as I have calculated, as well as other colleagues. It's the big part about the recessive clinical diseases where we need 2 mutations, one from the mother, one from the father, in one gene in order to have the disease such as the fibrosis, for example, cystic fibrosis and other diseases. Most genetic diseases are rare. It's as if you had a lot of minorities. I'm sorry for the mistake there, in my spelling. We make a collection of minorities. How you say minority A, B, C, D, etc., or religious minorities or other minorities, cultural minorities, geographical minorities. So think of our fellow people with genetic diseases as minorities. There are about 8,000 rare disorders, 75% of them are genetic, and we have about 300 million people in the world suffering from such disorders, most of them during childhood. And the biggest bet, the biggest problem to understand all genetic diseases are the recessive genes. We only know today 2,553 when we have 8 to 9,000 to get to know. And in order to find those recessive genes and find all these recessive diseases, well, it's not easy to find them in the industrial societies. In Europe and North America, we don't have a lot of children in families, and marriages in Western societies are not between relatives. We don't have consanguineous marriages. I don't like the way we call it in Greek. It's more like an incest, and I call it like relative incest. We may have, I don't know, an uncle with a niece getting married, etc. So the map of the world with these consanguineous marriages, there is a zone quite big from North Africa going to the Arabic states, and then Southeastern Asia, Pakistan, Bangladesh, etc., and the country with the biggest consanguineous marriages, biggest rate is Pakistan. About 6 out of 10 families are first rank cousins, and the population is 220 million. So if somebody wants to build on the knowledge of genes, that would be the populations for the research. So we started a big program that we call Swiss Pakistani Collaborative Consanguinity Project. And in Pakistan, we have 7 points of collection of samples with families with 2 or more children with the same disease. The children and all the normal children, and we studied the genes in order to find the changes. I'm not going to talk to you about the success of this program. The program is tremendous. We have found 60 genes from 600 families only. 1 out of 10 families makes us realize that there is a new gene. This is unheard. But I will talk to you about one family only. The joy of one, as I call it. If there is something we can do for one person in this world out of the 7 billion, then this joy, the joy of the knowledge, the joy of caring about this fellow human being is the biggest reward to a researcher. I will tell you the story of a child in Pakistan. This child, his family, is from a small village called Lachi in Pakistan, 50 kilometers from the borders with Afghanistan, an area which is very hot politically and quite risky. In this small part of the world, there's a family you can see here in the genetic tree, the father, the mother, first cousins. They have 2 children ill. They're blind, progressive blindness beginning from the age of 3 and the age of 6, 7, or 8. The back part of the eye, the retina, is totally destroyed. So it's a progressive retinopathy. So Selma, this girl, when we saw her, she had lost her sight, but not completely. She could see how many fingers, see the light, a few colors. We studied this family and we saw that the gene responsible for this hereditary disease in Selma and her brother is a genome that creates a protein which is a carrier. It carries a substance from outside the cell inside the cell. This carrier is SLC6A6. This carrier carries an amino acid called taurine. I don't know if you have heard of it. Anytime you drink an energy drink for energy, you have about 1 milligram of taurine. So this family has a problem of carrying the taurine from outside the cell inside the cell, and the retinal cell and heart cells need this substance, this protein, and since they don't have this normal carrier, they have this disease. The good thing in this family is that this problem in this protein is not 100%, but 10% works normally, 90% doesn't work at all, and 10% does it. It works as a carrier. So since this is done as a family, you have the family, the 2 cousins who married. The first child is older and totally blind. Selma is partly blind. So we thought that if we give a taurine to this family, if we give them a lot of supplements of taurine, since this defect in this carrier is 90% and not at 100%, then maybe we can treat or reverse the evolution, the progression of the disease. You see that we got permission from the bioethics committee in Switzerland to do that without control, since we only had one sick person. You see here the taurine, the magical medicine for this family. And after 2 years from this oral administration of taurine, well, the heart disease, because I forgot to tell you that apart from blindness, there was also a cardiomyopathy, like the athlete that you saw dying on the field. Cardiopathy was reversed. All indicators, the systolic and dilatation of the heart, diastolic rates of the heart were totally reversed. And now for the retina, I think it's hard to understand, but I will tell it anyway in simple words. Here you have a retinal electrical picture of this retina, let's say, and how it works. Where you see green, it's normal, and in the middle of the retina, you see this peak. There we see much better. This is normal. This is Selma. Selma is blue here. It's almost destroyed, and the center, at the center of her vision, she sees very little. After 2 years of taurine every single day at a dose that we calculated, her retina looks like this. It's not blue anymore. It's a green blue or green blue white, much better, and the center is about in the middle of the normal one. Selma, objectively, has now a visual acuity 6 out of 20, sees better. She can go to the market without assistance. It's the first time in the world that only one oral medicine, which is not really a medicine, it's a nutritional supplement, can actually not only stop but also reverse a retinopathy. And I'm saying that because when we understand the gene behind the disease, and if we understand the mechanism of the gene, then maybe after some time we can actually provide treatment. So, Ms. Michopoulou, by learning, getting to know the genes, instead of doing passive medicine, we can be very active. We can understand and prevent diseases, since everybody with are on the stage before getting sick. Everybody. I don't know how much time do I still have? As much as you want. We could be there all day listening to you. Thank you. So I will give you 1 or 2 more things, one for cancer and one for our origins. As we said, genetic diseases mutate all our cells, and cancer, on the other hand, is a genetic disease where mutations only happen in a few cells of our bodies. I will give you the story of my cells. When I was in the first cell, the zygotic, this zygotic was divided into 4, 8, etc. And every time we divide a cell, there are mutations that we call body mutations. So my cells now, that I am at the end of my 70th year of life, I have a lot of mutations that I didn't have when I was a child. or when I was in my first cell, these mutations are neutral. They do nothing positive or negative in the cell, but sometimes such mutations, such as this yellow one here, are pathogenic and cancerous. So a cell, well, in this progression of time, also accumulates certain mutations that can lead to cancer. But sometimes our organism expels them, but sometimes it doesn't make it. Very simply, this is how I can explain cancer. And here I'm showing you a study of the importance of mutations in cancers. Here you have the different organs of a woman. Where it's red, it's that mutations are very frequent for this cancer. Hereditary mutations for breast cancer, for example, have a very small percentage in the general population. Red here is really very fade, but all the changes that happen from the divisions of the cells are the ones that can cause cancer. And the environment, bad environment for the cell, creates mutations because of this impact. So lung cancer is very red because of environmental mutations, because when you smoke, then the cell is forced to mutate even further. It wouldn't have to do that without smoking. So mutations are the cause, but behind that, it's the environment that accelerates or affects the presence of mutations. We all know about coronavirus. We have two genes, 30,000 and 6.4 billion. There's a war. Who will survive? Each is only interested in their own lives, in their own survivals. As people, same with genes. This is the theory of selection, of natural selection. I need 2 more minutes to talk to you very quickly about the genome, which is also a history book, personal history book, and of our kind. Because in each generation we have mutations, and most of them stay in our DNA. So when we read our genome, we can actually see the history of our genes in evolution. We know by analyzing genes of thousands of people in the world, our historic trip of Homo sapiens started from Africa, South Africa, in the years, thousands of years that you see here. This may change in the future with a better understanding of the speed of changes. Let's say that we started being on this planet southern of Africa, and after 50 to 60,000 years went to the Middle East and then conquered the world. In Europe, 40,000 years, and then North America from Alaska, 15,000 years, and the fresh populations are the ones living in South America. Now, if I read my gene, can I read my origins? Yes. I can read my mother genealogy, because within my cell, apart from the nucleus with the gene that we said so far, there are some, there are the mitochondria that are the energy production plants in the cell. They have a smaller gene, 16,000 letters. And the characteristic of this mitochondrial gene is that this is transmitted from the mother only. Mother gives it to the children, and because this DNA doesn't come from the sperm. So my mitochondrial DNA, I got it from my mother, who got it from her mother, etc. So if we study my mitochondrial DNA, we can see, we can go back to my mother's tree. I come from Africa before 180 years, 1,000 years ago. Can we see father's tree? Yes. We can record it from chromosome Y, sorry. You know that women have two X chromosomes and men one X, one Y. The Y, we only get it from the father. So my Y chromosome, I got it from my father, he got it from his father, etc., till the beginning of our kind. If we do this again, we can see that my Y chromosome came from the, South Africa, again, more or less same at the same time. So we can read our story, not only what happened in the recent 5-10 generations, but in the depth of time from the beginning of the humankind. So gene or genomatic medicine is not just joy. There are many issues involved, ethical, social, and legal. I'm not going to analyze them in detail, but one of them is about individuality, the tyranny of knowledge, the therapeutic gap, genetic perfection, intellectual property, and gene correction in the reproductive cells. For most of them, we can make laws and protect individuals from concerns. Apart from two, the tyranny of knowledge and this therapeutic gap, the tyranny of knowledge, let me tell you about that, was very well explained by Sophocles in Oedipus. Sophocles, who lived about 450 years before Christ. In the third scene, if I'm not mistaken, asks Oedipus, Tiresias, Oedipus, without knowing, kills his father and marries his mother. They have 4 children, and at some point they understand that something bad happens to his family and knowledge, and asks to the blind medium or genetist, what is the bad thing that has come upon my city, Thebes? And Tiresias asks, what do you do with the knowledge if it does not really, if there is no use for it? It's the complex of Tiresias, or it is the therapeutical gap, which means that the diagnosis runs faster than treatment. And for many people, they choose not to know the risk involved, and many times when we cannot do anything with the knowledge, we prefer ignorance. But a big part of the population wants to know in order to program the rest of their lives. And I will end with this distinction of gene distinction. We should not classify, categorize people with some gene diversity. I'll give you an example from Dr. Robert Ritter in Berlin. In his report in the Ministry of Health in Berlin on 31st January 1941, he wrote, with the money that you gave me for my research, I recorded 23,000 cases of Roma. And then we read in a Nuremberg trial documents, out of the almost 21,000 Roma entering Auschwitz camp, look at the number, 23,000, 21,000. 3,046 one were transferred to other camps. Why was that? Because they said yes to becoming sterile forced. The rest died of diseases, hunger, or poisonous gas. And why do I say that? As geneticists, we have made a lot of mistakes in the past. But now we have a better knowledge of history, of medicine. We need to consider all that and be very careful in the way we proceed, and also accept diversity as a gift of nature, because everyone who is diverse is finally same as all of us. I would like to thank all my patients and their families, and close with what Antonio says in Shakespeare's Tempest. What is past is a prologue. So our past is just the beginning. Thank you for the interest. Thank you, Ms. Michopoulou, for this invitation. Professor, thank you so much. Now, what can I say? What can I comment? What can I ask? You've said it all. If I could, I would bring you here every day for a lesson. There's a question from the audience. Now, about women, we don't have our Y chromosome. Can we see our family history? Yes, if you read the gene of your father, you will find the Y chromosome and follow it. Now, seeing the athlete just dropping dead, if he had made a genetic control, maybe he would live now. Everything is written in our genes, but do we want to know what life reserves from us? Because people usually say, I don't care, I don't want to know, whatever comes, let it come. So how do we convince the people to go and check themselves? Well, there is no rule about all that. For the athletes, as you said, if they knew their genetic predisposition, they could have a fibrillator in their heart, for example. They would avoid sports with a lot of practice, with a lot of exercise, thus, improving longevity. But there are people who have, a theory, a stance vis-a-vis life. They don't want to know, and there are others who want to know everything. Geneticists and medical doctors, they go accordingly to what people want. It's a free choice, but it has to do with the knowledge of the object, and many people don't have this knowledge. If all pregnant women did this genetic test, then maybe a lot of genetic diseases could go away. On the other hand, everybody has the right to life. What is your position on that? Well, same as the previous one. Each family have the possibility to decide on their own, depending on the history, credos, education, theory of life. And finally, when there is a disagreement, and I see that every day, between parents, it happens frequently. Usually the mother is the one having the last word. We need to close, but let's make a myth collapse. The vaccine mRNA will enter our DNA and make us zombies. No, RNA is a very unstable molecule. It enters the cellular plasma, gives an instruction to create a protein that creates antibodies. RNA vaccines are the best recent tool that we have against the virus. And if I have to end this talk, I will end it by encouraging everyone to be vaccinated. Last question. You investigate the mystery of life every day of your life. What is the most important thing in our life? Man, humans. We must be and remain humans all the way. And of course, science goes hand to hand with humanity. Of course. Thank you very much. It was an honor and a great pleasure. We expect you to be here again next year in the Athens Science Festival, hopefully live, to give us all the new things about your research. Thank you again. Bye.
Την επόμενη χρονιά να τα ξαναπούμε.
Καλή δύναμη, καλό κουράγιο και καλή επιτυχία.
Ευχαριστώ.